Our first speaker doesn't need any introduction. He's our big boss. Jens Nielsen, who's going to tell us something about the systems biology and identification of novel targets for metabolic engineering.
Thank you very much, Maciek. First of all, let me thank Ryan for inviting me here to give a talk and for setting together a fantastic conference, a very good program. I'm going to talk about following up from what we heard about this morning, use of systems biology in metabolic engineering, and set the scene very fast. Basically, the objective is, of course, to develop this biorefinery concept where we develop efficient cell factories for conversion of biomass-derived feedstocks into fuels and chemicals. We have been talking a lot of that, but of course the real enabling technology is developing of the cell factories as the catalyst in these processes. So we work primarily on yeast. Yeast is used already widely. We have heard several examples already at this meeting here. It has some advantages being extremely well characterized. A lot of information is available, both at the industrial scale level, but also at the laboratory scale level. And it's, as I mentioned, is already used widely, but also a number of new processes are in the phase of being developed. So in my group, we are working on developing yeast as a platform organism to produce a range of different products. Some are listed here. They're spanning from high volume, low value added products as like biofuels to the other end of the spectrum. And you can say the scientific focus here is to look if we can really engineer and change yeast metabolism. Can we make it sufficiently flexible to produce these different compounds at high levels? And in connection with that, we are working on developing enabling tools from synthetic biology and systems biology to, you can say, speed up also this development process. So when we talk about the metabolic engineering, and this session here is called Design, Build, Test Cycle, which I guess is really the essence of metabolic engineering. We're talking about typically the use of modeling for the design. We will then construct strains, and particularly this field here has really, I think, particularly with yeast, faced a tremendous development in recent years. We've been able to do this much faster using a number of different synthetic biology tools. We will then typically evaluate the strains in fermentation, and then we will often do a very detailed phenotypic characterization, maybe not in each round of metabolic engineering, but if we want to get a better understanding about what is really happening as a result of the genetic modification we performed. And it's those two components here that are really taking advantage of developments in the field of systems biology, both in terms of modeling, but also in terms of analyzing and integrating these different types of omics data. And I'll come back and talk more about particularly use of omics data and genome sequencing for phenotypic characterization. But let me first talk a little bit about development of platform strains and also an example of a traditional design, build, test cycle. So as I said, we are interested in developing platform strains that efficiently can produce and provide specific precursor metabolites that can then be used for production of a number of different products. And acetyl-CoA is an interesting precursor metabolite because it's used for the biosynthesis of a range of different products that have industrial interest. Isoprenoids we already heard about, PHBs, 1-butanol, 3-hydroxypropionic acid, different types of lipids, and so on. And of course, in Saccharomyces cerevisiae, we are facing a challenge because we would typically like to implement and reconstruct these heterologous pathways for these products here in the cytosol. So that means that we'll have to get acetyl-CoA produced efficiently in the cytosol. And so we developed and are working on developing tools like this. There's been some beautiful examples also recently in a paper from Hu Min Zhao about also targeting this approach here. But besides, of course, efficient provision of these precursor metabolites here, we would typically also like to have a list of preferred phenotypic traits for these strains. It can be that they can go at high temperatures, low pH, they should be tolerant to different types of alcohols and so on. And this list can be very long, and these will also be traits that we would like to integrate in these platform strains. So first a little bit about acetyl-CoA platform strain. So we generated what we call a block push-pull strategy a couple of years ago. So basically by increasing flux down towards acetyl-CoA in the cytosol, but at the same time also pulling in ethanol that are produced typically at high sugar concentrations during growth on yeast by overexpression ADH2, but also overexpression ERK10, in this case here for products derived from acetoacetyl-CoA. And hereby, with this strategy here, which we combined also with deletion of MLS1 or SID2 for blocking the use of acetyl-CoA for other routes, for other processes in the cell, we could increase the production of isoprenoids 3 times, PHB 80 times, and 1-butanol 5 times. So this was an example of a generic technology, basically a generic platform strain where we can hook on different heterologous pathways and in all cases have an improvement. We were interested to exploit this further for production of 3-hydroxypropionic acid. This is an interesting platform chemical, and there are several initiatives going on industrially on producing this. We were looking into different pathway routes for this. There is the so-called beta-alanine route here. There is the what we call the malonyl-CoA route, where you basically go from malonyl-CoA via malonate C-maldehyde to 3-HP. There's the glycerol route here. The glycerol route is fairly complicated to reinstall in yeast because it is involving a vitamin B12-dependent dehydratase. And finally, there's the lactic acid pathway, where not all the enzymes have been identified. We were interested, as I said, to use our acetyl-CoA platform and see if that could work, so therefore we were looking further into the malonyl-CoA route. Conversion of malonyl-CoA to 3-hydroxypropionic acid can happen either by a multifunctional enzyme, MTR1, or a 2-step pathway going via malonate C-maldehyde. And we evaluated first enzymes from different sources, either the one-route step or the double step here, and saw which ones was effective and most effective, and MTR1 turned out to be actually a quite efficient enzyme in taking malonyl-CoA to 3-hydroxypropionic acid. Just by simply expressing this single enzyme, we can produce about 100 milligrams per liter of 3-HP in a standard yeast fermentation from 20 grams per liter glucose. But if you look a little bit further on engineering and improving the flux down to 3-hydroxypropionic acid, you can see that we are facing a couple of challenges. Of course, there is the supply of acetyl-CoA, but also further malonyl-CoA, as well as also we need to balance the redox. This enzyme here requires 2 NADPH down here, so we need also to ensure efficient provision of NADPH to get this going. So we therefore broke this project down in several rounds of metabolic engineering to first look at the first precursor here, acetyl-CoA, then later on to see how we efficiently could produce this one, and how could we also cope with the balancing of the NADPH. So first, for the acetyl-CoA, here we use this push-pull block strategy that I mentioned before, and you can see here simply by overexpressing MTR1 alone gives about 100 milligrams per liter. When we combine it with this push-pull block strategy here, we can increase the strategy. But in this case here, really overexpression of ACC1, acetyl-CoA carboxylase, is also quite important in order to pull the acetyl-CoA to the malonyl-CoA. But you can see these simple strategies here really are quite effective here, giving more than a 2-fold improvement in 3-HP production. We then were looking also on provision of NADPH. Here we were looking into an enzyme that we have been working on before in connection with improving ethanol production, namely GABN, which is a glycolytic enzyme from a lactic acid bacteria that bypasses 2 enzymes in the endogenous glycolysis pathway of Saccharomyces cerevisiae. And by overexpression GABN alone actually can give more than a 4-fold improvement in 3-HP production, really pointing to most likely a kinetic constraint in provision of NADPH for 3-HP production. Now, I mentioned that we overexpressed ACC1. ACC1 is a little bit of a special and also a key enzyme. It's highly regulated in eukaryotic cells. It is phosphorylated by SNF1, which is a key protein kinase that is in responding to particularly ATP limitations in the cell. And SNF1 has been identified earlier to phosphorylate as ACC1 at this position here, 1157 serine. But we identified another putative site based on analysis of the sequence. And what we did was that we removed these two phosphorylation sites, and we could see that that alone gives a more than a threefold improvement in 3-HP production together with expression of MCR1. Removal of each of the individual sites here also has a positive effect, but the double removal is the largest effect. So here we have an example where we have increased the supply of acetyl-CoA. Here we have improved the efficiency of the further conversion to malonyl-CoA, and also we looked into the effect of NADPH supply and what happens if we combine these different strategies. You can see we, in combination, can get a 10-fold improvement in 3-HP production, so we can surpass more than 1 gram per liter when all these strategies here are combined. So as I said, this was an example of a typical how we often work in metabolic engineering. We go through these cycles, we find the different type of targets that we then improve through rational design cycles. But what do we do when we're talking about these more complex phenotypic traits? How are we going to come around and improve, for example, temperature tolerance or tolerance towards alcohols? It can be butanol or higher alcohols. This is typically more complicated because we do not know exactly what are the mechanisms for driving these phenotypic traits, so we will not know where we should set in. And here comes also another issue that is quite complicated, namely the impact of regulation. And so I think many of us working with Saccharomyces cerevisiae have sometimes been a little bit frustrated about to see the rapid speed of advancement you can do metabolic engineering in E. coli, where you typically can relatively easily redirect flux, whereas in yeast it is typically much more complicated. We were interested in the context of that, beginning to get more understanding about what is actually regulating, controlling the carbon metabolism in Saccharomyces cerevisiae. So we were therefore looking into yeast transcriptional regulatory network. And one thing that was a big surprise for us was that we found when we built these networks here, where we basically identifying a transcription factor, if it's regulating expression of another transcription factor, and that is again regulating another one, we can look into these kind of structures here, networks here, and we found that actually only 17% of all the transcription factor needs to be controlled in order to control the whole network. That corresponding number for E. coli is 64%. So that means that really only by hitting a few key transcription factors we can actually regulate the whole thing. In other words, it also means that this is highly connected, this network of transcription factors, and particularly because you have a situation with many transcription factors regulating the same genes, you have a lot of redundancy and overlap here. One other interesting finding was that the yeast transcription regulatory network is actually not scale-free. It doesn't have direct implication as such, it's just an observation. But you can see, compared with the Regulon DB, which is the corresponding E. coli network, it's follow a scale-free model here, whereas the different yeast models here, either the two Harbinger networks here or the yeast tract, really deviates from this and comes closer actually to a random network simulated here. And one of the reasons for this large variation of the yeast network is actually there is a very large loop similar to this one up here. Here is just shown with 3 transcription factors, but there's a very large loop encompassing more than 78% of all the transcription factors in yeast. One could speculate about the evolution of these loops here. It's very likely that that can give increased stability. But with permutations or changes, for example, in the kinetic landscape, it can actually also easily shift over and give a very unstable system. And this is, of course, one of the causes that could be the causes also of cancer, that a few mutations can actually tilt the kinetics of such networks that have very large loops. We put in here also regulatory networks from human and mouse shown here, and you can see they actually follow more the yeast networks. So if you look at the structure of these networks here, you can see hierarchy is a difficult word to use in this context here, but basically the coli network is more hierarchical in the sense that the one key transcription factor is kind of sitting as a supercontroller, whereas in yeast you have several sitting at the top here, and also they are cross-talking to a very large extent. So these are, as I said, some of the challenges. We were then interested to see also, can we identify some key environmental cues that regulates or fixes some expression of some of these different transcription factors. And so here we were looking into transcription data from more than 200 different experiments, all performed in chemostat cultures, where you have a very tight control of the environmental conditions. And we were then looking into specific environmental changes, could, for example, be aerobic to anaerobic shift, or it could be from carbon to nitrogen limitation, and so on. And we were then looking in to transcription factors which do not have their own expression changed in variation for these two conditions, as indicated here. So the genes that they regulate are significantly changed between these two conditions. So these are likely to be transcription factors that are kind of getting a signal from the changes in the environment, and is changing also the regulatory cascade in connection with that change. And not surprisingly, maybe in the context of the very tight and the very large loops we are finding, we find very few of these transcription factors that are really driving specific environmental cues. Okay, so this clearly shows that in Saccharomyces cerevisiae we have very, very tight regulation of metabolism, and if we're going to go for these engineering of these complex traits, it may be very, very difficult because, of course, years of evolution have pushed it into a certain phenotypic type. So this brings to use directed evolution to basically both learn about how we can unravel this regulation here, but also at the same time pick up new strains that have these specific phenotypic traits. So we were looking into tolerance to butanol. There's a lot of interest in producing butanol commercially, of course, but also butanol is interesting because it's extremely toxic. So whatever we could do in order to improve the tolerance towards butanol is likely to give us new insight into basically how Saccharomyces handle these kind of stress here. And so what we're doing here is that we're doing serial shake flask transfers typically with increasing ethanol concentration, with the objective to basically, you can see here for the wild type strain here, the drop in specific growth rate with increasing butanol concentration. The objective is, of course, to push this up and find mutant strains that have a higher specific growth rate at a higher butanol concentration. A little bit to our surprise, it was very difficult, well, basically impossible, at least at the duration of our evolution experiment, to pick up mutants only by adaptive laboratory evolution. It simply seems that we need more mutation rate, higher mutation rate, in order to find mutants that can cope with butanol. So therefore, what we did was that we added mutagenesis. So we added random mutagenesis, UV mutagenesis, and combined that with adaptive laboratory evolution in order to pick up clones. And eventually we could pick up 10 clones that had increased specific growth rate at a butanol concentration approaching about 1.2%, some with a little bit lower resistance to it. And we could, you can see here, these are some of the clones here, characterized here, compared with the wild type. They have an enriched specific growth rate up to, in this case here, also 1%. And we then performed a very detailed phenotypic characterization of these 10 strains. We did, of course, this in extreme, well-controlled bioreactor experiments where we could clearly quantify what was the effect on the specific growth rate, specific glucose uptake rates, different types of byproduct formation rates, and so on. We performed genome sequencing of these strains, we performed transcription analysis, and also we performed endometabolome, including lipidome analysis. So I'm not going to have time to go through this whole analysis here. It's a very large data set we generated here, more than 200,000 data points. But one thing, one key lesson though that came out of this study here was that in order to cope with increased butanol concentration, you really need to reprogram pretty globally. And many different processes in the yeast cell has to be reprogrammed. And that's probably consistent also with the fact that we could not find mutations just by having an adaptive laboratory evolution experiments. Maybe we could have done that if we went on a long time, but I should say that we actually performed AIChE for more than 6 months in that process, so that may have to be carried out for a very long time. So what we see here is that we see effects on ROS, we see effects on efflux pump, we see changes in the cell wall composition, we see changes in the pentose phosphate pathway associated with NADPH production, glycogen trehalose effect, we see effects on beta oxidation, and finally we also see effects on vacuole acidifications. And when I say we see all these effects, some of these effects are directly measured, some of them are inferred from the changes in transcription as well as also changes in genomic mutations. If we just take one of these, detoxification in response to reactive oxidant species, we can see here that we have a number of effects here where we have upregulation of the oxidative phosphorylation and also ROS detoxification pathways. So we really seem to have processes increased that involved in scavenging of intracellular radicals. And this is, we believe this is very much due to probably conversion of the butanol to aldehydes that can be very toxic for the cell. If we looked at efflux pumps, it was maybe surprising to see that there's not just a general upregulation of efflux pumps. Some efflux pumps have increased expression, as marked here red. Others are actually reduced expression. Interesting enough, we also see increased expression, or actually altered expressions, some increased, some decreased here of these yeast translocases that are basically involved in flipping the different phospholipids from the inside to the outside face of the membrane. And so it is believed to have an impact both on the membrane fluidity but also on the membrane curvature. So that is also affected. Finally, to mention here, as I said, we did see some effect on fatty acid composition. We see that clearly as a trend towards longer chain fatty acids, but also a trend with more double bonds inserted in, or in this case, just a single double bond. So we see also upregulation of both the elongases here, but also of the desaturase, delta 9 desaturase that inserts a double bond in C16-0 and C18-0. So and that the upregulation of these enzymes was consistent also with elevated levels of longer chain fatty acids, the C18, 0 and 1, but also more saturation of these. So this was a very complicated case. When we look into the genome sequence analysis, we do find a number of different mutations appearing here, and we're currently evaluating some of these single point mutations. We're not hoping really for magics here, I should say, because there are so large number of mutations typically, and with this global rewiring towards butanol tolerance, that's probably difficult to really find key targets to pursue a kind of the silver bullet that will improve tolerance towards butanol. So, but let me end with another story where we will look into adapting yeast to grow at higher temperatures. And here we could say we do know quite a lot. We do know a lot about the heat shock response, and could we not just target some of the components of the heat shock pathways and, for example, overexpress these or take them out? This has been attempted to do, but it doesn't typically give the long-term effect. So this is a process that has typically been installed in evolution to cope with typical shock moments and just kind of survive for a period of time until the temperature is low again. So generally what has been observed from previous studies is that there's very little link with the heat shock response and ability to grow at high temperatures. So again, also in this study here, we did adaptive laboratory evolution. We selected strains that could grow at 42 degrees Celsius. We then isolated these clones and characterized them in bioreactors. We performed genome sequencing and also, again, multi-omics type analysis. And we did find a number of strains In cereal shake flask cultures, we identified 9 clones in 3 parallel evolution experiments shown here that have increased specific growth rate compared with the wild type. These are from shake flask cultures. The first line here indicates the average from the 3. We characterize these in bioreactors here, and you can see we almost managed to double the specific growth rate in bioreactors. We had impact also on the specific glucose uptake rate. We can see that the biomass yield was slightly improved, indicating also strongly that the energetic efficiency of utilization of glucose was also improved in these strains at 40 degrees. From genome sequencing analysis of this, we found very much in 3 from one of the flasks here, and 3 of the clones identified, and 2 from another flask series here, we found duplication in part of chromosome 3. This has been reported earlier to have an association with the ability to grow at higher temperatures. So we were, of course, interested and curious to pursue that. But interesting enough, it turned out that in one of the other flasks, where we isolated 2 clones from, there was not this duplication. So that hinted, of course, too, that there is not, it is associated with this adaptation to grow at higher temperatures, but it's not really an essential requirement. We identified also a number of single nucleotide variations in the different clones. We found some that were unique and actually a single gene that is overlapping in all, that is ERK3, and I'll come back and talk a little bit about that. We also found ATP3 here to be in several of the clones identified. But first, let's look a little bit about this chromosomal 3 duplication here. If we look into which genes that are in this segment here on the chromosome, many of these genes are also having higher level expression, and many of them are involved in the cell cycle and the duplication. So we see that there could be a link by having upregulation of these genes through chromosomal duplication, and that actually impacts. We also see genes affected on stress tolerance also in this segment here, but also a number of other genes in these segments. But as I said, one series of the strains did not have this chromosomal duplication, so we do not believe that this is really the key driver behind this. So we were therefore looking into the single nucleotide variations. It's quite interesting that a relatively large fraction of these, 26%, results in appearance of stop codons in the genes, and 66% of these stop codons appear in only these 2 genes here, ATP3 or ERK3. And therefore, it was, of course, particularly interesting to look into these. And when we delete or insert the corresponding stop codon for ATP3, we do not have an effect. We do not have an enrichment. Actually, it's detrimental. ATP3 is a key component of the F0, F1 ATPase, and it basically takes out respirations and also really the ability to grow efficiently. Interesting enough, if we introduce the stop codon identified from ERK3, you can see we can recover about 85% of the phenotype in terms of the specific growth rate at elevated temperature compared with the temperature-tolerant strains. And it's, of course, interesting to see what ERK3 is doing. ERK3 is one of the, not surprisingly, component of the ergosterol biosynthesis. It's further downstream. And one thing that we observe in this strain here is that we get a shift from presence of ergosterol to phacosterol as a key component. Ergosterol being a linear sterol, phacosterol a bended sterol that may have this effect of changing basically the membrane properties in the cell and hereby conferring this improved tolerance to higher temperatures. This is somewhat consistent also with an upregulation of all the, practically all the sterol or the ergosterol genes, as well as further back to the mevalonate pathway. This pathway is highly regulated at transcriptional level by the levels of sterol, ergosterol in the membrane. And if we're basically removing ergosterol, we take away this regulation and we get our upregulation of the whole pathway. One thing that was interesting, less interesting, I should say, from an application point of view, but as an observation, was that the thermotolerant strain all had a trade-off in the sense that they could not respire at 30 degrees. So basically, they have lost their respiratory ability. This is, of course, consistent also with this point mutation picked up in many of the strains in ATP3, but actually several other genes are associated with this. So it really seems that when we are evolving towards higher temperature, growth at higher temperatures, we are taking away respiration first, and it's maybe further downstream that this really driving mutation, ERK3, appears. So we were, of course, interested to see what happens when we insert this stop codon in ERK3. Do the cells also have this trade-off here? And very interesting finding is that it is not the case. So we can actually get the ability to grow at higher temperatures with a single introduction of a single point mutation, and we do not have this trade-off in terms of respiratory growth, as shown here in the light blue line here. This is for the wild type grown at 30 degrees. These are the thermotolerant strains. You can see they grow slower, and they don't have the dioxic shift growth. So with that, let me come to my conclusions here. The rational design build test cycle works very well when we're talking about reconstruction of pathways and improving precursor and cofactor supply typically. But it's more difficult, of course, to apply when we talk about improving complex traits. And here I hope I demonstrated to you that AIChE is a very powerful tool. And in some cases, we can really find single key driving point mutations that can then be integrated in a clean strain and thereby confer the phenotypic trait that we are interested in. So let me thank the people here. Several of them are here at the meeting also. Yun, Jitchen, and Verena worked on the 3HP story. Anastasia has been working together with Yun and Kanokan on the acetyl-CoA platform strain. Tobias did the analysis of the transcription factor network. Antonio on the butanol story. Luis on the temperature story. And collaborated with several people here. Dina has been closely involved, and who's here in the audience also been closely involved in the temperature adaptation story. Thank you very much for your attention.
Thanks for a great talk. We have time for a few questions.
Yeah, Jens, very nice talk, interesting results. I have a question regarding the transcriptional regulator network of yeast. You mentioned the network is somehow quite different from E. coli, and I was a little bit surprised to know you said 70% of the genes control the whole regulator network in yeast, and on the other hand, 64 of the genes need to somehow disrupt the E. coli transcription network. How you define the 17 or 64 genes which control the network? In my understanding, actually, E. coli actually has just a few global regulators. If you knock out them, you will destroy all the network.
Yeah, thank you. Basically, you could say you can look at it from these simple networks here. So in this case here, you can control the whole network by targeting transcription factor A. So basically here we can target the network by just one transcription factor. In this case here, we would have to have two input nodes here. In this case here, because it's this large circular loop, we can basically hold the whole network by targeting either A, B, or C. So you can have a score for this where you basically evaluate for each of the transcription factor how can you activate them by targeting a certain amount.
Do that correspond to the so-called global regulators or different?
Yeah, you can say it's a little bit difficult to have a discussion about these terms because we talk about global regulators in E. coli, and of course a very typical example of global regulator is CRP, who is at the top of this cascade here, and which can really target a lot. But basically, what this also means is that if you regulate any of these transcription factors down here, you don't really regulate any of the other ones, right? So therefore, hierarchical regulation is a term that kind of needs a very solid definition before one has a discussion about whether there's more hierarchical regulation in E. coli or than in Saccharomyces.
Okay, thank you.
Yeah.
Okay.
I actually have a question. So in your butanol tolerance adaptive evolution, I noticed in your results that your evolved strain grew slower at the unstressed conditions. And we actually did similar evolution experiments in my lab, and we found the same thing. So can you comment if you think that's a necessary, if those two things necessarily go together, or what's the mechanism behind that?
I cannot tell you about what mechanism is behind it, but I think it's something we generally find is trade-off towards the kind of the reference conditions. We found that in our study on evolution towards improved galactose, growth on galactose. We also found a trade-off in terms of growth on glucose. And often you talk about two different types of evolutionary trade-offs here, either accumulative mutations or pleiotropic mechanisms. And often I think you would find this pleiotropy here, where you basically, the mutation that are beneficial at condition A is the one that causes a trade-off also. That doesn't seem the case in the case of temperature here, because we are targeting basically something that is not so detrimental, a little bit change in the membrane structure. I'm pretty sure that we may have some trade-off mechanisms, but probably not in terms of growth on the temperature case.
Okay, let's thank the speaker again.